Welcome back, everyone, for the afternoon main stage segment. This is going to be so good. So hold on to your seat. We have a series of talks. Our first one, I don't even need to give an introduction to. A true legend and pioneer in synthetic biology. It is a pleasure to introduce Jason Kelly, who's going to be speaking about making biology easy to engineer. Let's give it up for Jason.
All right. Good to see you all. Okay. I think we should have some slides up in theory. Okay, yeah, so I'm going to talk to you today about what Ginkgo can do for you. It's written on the shirt. But before I get into that, just as a reminder, Ginkgo operates as a platform company. So think of us a little bit like an Amazon Web Services, right? So people outsource to Amazon, to their infrastructure to build their applications on top of it. And that's really what we're doing at Ginkgo when it comes to synthetic biology technology. And we want to really hear from all of you today and at our booth over there about what it is we can do to help you, all right? Before that, though, I do want to take a minute to talk about what a big year 2022 was for synthetic biology, starting with the return of John Cumbers to leadership here at SynBioBeta. I want to give a real round of applause to John for putting together this conference and really ultimately being really a pillar member of building the community in synthetic biology. We're very lucky to have him back. The other thing you might have noticed, there's an executive order from President Biden here in the US in September of last year, and I'll read this part here. We need to develop genetic engineering technologies and techniques to be able to write circuitry for cells and predictably program biology in the same way in which we write software and program computers. Hell yes. Okay, this is something that people in this room have been really looking to see from leadership in the country and broadly for a long time. It's really awesome to see it. About a week after that, there was a big meeting in the White House. I was there, Gaurav at Solugen, Jennifer at LanzaTech, and then basically the heads of all the major agencies here in the United States, Health and Human Services, Department of Energy, Department of Agriculture, DepSecDef. Jake Sullivan, National Security Advisor, was leading it, and he basically said, hey, you need to bring more biology into these departments. How are you going to do it? Okay, and so I think you see this momentum. Also, we were lucky to hear yesterday from Renee. So about a couple weeks after that meeting, President Biden came to JFK Museum in Boston, announced the launch of ARPA-H. Okay, ARPA for Health, and no surprise, Ray Wegerson, shown in green there, selected to lead that. Card-carrying synthetic biologist, right? So Renee used to run the synthetic biology programs previously at DARPA. So again, I think this is indication that this technology is ultimately underlying sort of strategic technology assets here in the US. The other thing, just to put in a plug, tomorrow morning I'll be on a panel with Michelle Rozo and Rai Barkot here at SynBioBeta to talk about a new commission, National Security Commission on Emerging Biotechnology that I'm chairing. Michelle is the co-chair. This is based on a similar commission on AI 3 years ago that Eric Schmidt was the chair of. All right, so think a little bit like in synthetic biology, we're sort of where AI was 3 years ago, all right? And again, this is to have the US make sure we're being competitive when it comes to this technology internationally, and then also to look at the national security defense implications of synthetic biology, things like biosecurity. And we're actually doing a fair bit of biosecurity at Ginkgo, and I'm not gonna spend a ton of time talking about this today, but just to get it on your radar, we run a program with the CDC. We collect wastewater samples from planes, sequence if there's a positive. This was looking for COVID, now updated also for flu. We caught the first cases of BA2, BA3, Omicron, sequenced in the US, 3 or 4 weeks before they showed up in hospitals. We rolled this program, expanding it into Qatar, Rwanda, Ukraine. The idea here is ultimately to have a radar, kind of a global weather map of how viruses are moving around so that we can be more prepared. A year ago here at SynBioBeta, Eric Schmidt was on stage, and I'll read the quote, which I really liked from his talk. He said, it's time to take the work of the last 10 or 20 years in synthetic biology and make it scale to a global phenomena that's somewhere between $4 and $30 trillion of economy. You all, as you all sitting there, will do that. That's why I'm here. And this is really, I think, this is what it's all about. Like ultimately, we are trying to develop applications that have impact in the real world. And Ginkgo's view is that we can be a platform to enable all of you to do that, right? And the reason we believe that is that biology shares a common code. In other words, DNA is common across all of biology, and thus we believe it's common across all markets, you could have a platform that would make it faster, more reliable, and cheaper to program that code. And that's what we're trying to accomplish at Ginkgo, and that's why we're excited to be with all of you today. I will note, a lot of people did not think that would be feasible. Right? So for example, the automation you would need to work with mammalian cells is too different than what you would use for microbial cells or plant cells. The machine learning models and data science you would use for an agriculture project is too different from what you would need for a pharmaceutical project. I think we're proving people wrong on that front at Ginkgo. It's a list of customers, some of the customers on our platform. Up at the top, you can see larger companies, pharmaceutical companies, Merck, Novo Nordisk, just announced a deal with Boehringer in the last couple of weeks, Biogen. Industrial space, Sumitomo Chemical, Givaudan's largest flavor and fragrance company, just about a month ago, Solvay on the platform, European chemical company, and in agriculture, big three ag companies, Corteva, Syngenta, our longtime customer Bayer, all leveraging the platform. But more importantly, on the lower half here, you can see many of you in this room, right? So, we work with Genomatica, Motif, Bolt Thread, Synlogic, Antheia, Archaea, Procarium, Optivia, Sensible, Ayana, Alonia, Humblebee, Lygos, a lot of the companies that are going to disrupt these industries in the future and are in this room today are leveraging Ginkgo's platform. And importantly, the rate that people are coming to the platform is going up. So in 2022, we increased active customer programs 60%. We increased the rate that we're adding new customers by 90%. And this is the sort of thing that is, investors get excited about. That's good, right? I'm happy about that, to deal with investors a lot. But it's also something we think our customers should be excited about. And the reason is Ginkgo's platform gets better with scale. Right? Again, I'll draw your analogy to that, like cloud computing or making semiconductor chips, right? Like the more business on these automated scaled platforms, the more efficient they get and the more value they can give to customers. And in particular, what we want to offer is less risky cell engineering that takes less time. In other words, you get to a product more quickly, and that costs less. Okay, I'm going to talk more about that in a second. But our belief is every time a new customer signs up, you're more likely to get those three things. And so you should be excited to see new customers on Ginkgo's platform. If you're one of our current customers or if you're a potential customer, it should get you paying attention. Okay, so I want to spend a minute. We do, again, I will highlight, just as a reminder, we don't develop our own products at Ginkgo. We are a services platform to enable your products. So what we do is we spend time talking to customers about what they would like to see, why are they choosing to outsource to our platform. And just to be extra clear about this, the status quo today across all of biotechnology and all of these industries is to do this work in-house. Right? If you're going to do a genetic engineering project, you're going to build a lab, you're going to buy a bunch of equipment from Thermo, you're going to set up, and you're going to get a bunch of R&D scientists, and you're going to get to work. And our view is that work would be better outsourced in partnership with your scientists, working with Ginkgo scientists and our automated infrastructure to do that same work. That's the fundamental shift we're aiming for. So why did Novo Nordisk choose to work with us? Well, Brian Van Dahl, who heads up global research there, technology, said, large-scale data sets coupled with AI is opening up a greater opportunity space within biology. We no longer have to limit ourselves to the questions that can be addressed by traditional research methods. And so I think Brian's quote is good for sort of the top two reasons that I see why people are choosing to outsource. The first is you get more data per research dollar. And this is really critical as we're entering an era where data is being used to train these sort of big, large machine learning models and so on. Second, it's not just your data, right? By leveraging Ginkgo's platform, we're actually accruing data across these now nearly 100 R&D projects on the platform so that you get access also to data happening across the industry. And I talked about that the other day on the pharma panel. I think all of us trying to do this ourselves is one of the reasons that this work is so difficult. And so on Ginkgo's platform, we're able to pool more of that IP and data across projects and companies. And then the bottom three reasons, which I'll spend a minute on, I think are particularly important for startups. You can launch work more quickly on Ginkgo's platform because it already exists. Second, you don't need to build a freaking lab, so you can save money on laboratory CapEx. And third, you can replace sort of a fixed cost, like you're always spending the same thing on your research infrastructure, with a variable cost. When you need more, you spend. When you need less, you spend less. Okay, and that can, add a lot of value. All right, so I'm going to talk about each of these in turn. So the first, you get more data per R&D dollar. It's a nice video. So Ginkgo acquired a company locally here in the Bay Area, Zymergen, last year. You can see some of their technology in these videos. And so one of the things that we're excited about is that the very latest technologies in automation, we think will drive down the cost of doing this research for all of you. If you are developing a AAV gene therapy or a fragrance molecule or an animal-free meat product, you should not have to be a world expert in laboratory automation. Right? You have a million other things to worry about in developing that product, right? And so we want to take that on. We will pay attention to the bleeding edge of this technology. We will integrate that at Ginkgo, and you will get to benefit, right? And again, we see this, the CSO at Novo Nordisk, Marcus. It was Ginkgo's ability to rewrite genomes to engineer new bespoke biological systems, so sort of large-scale engineering. Biogen, we did an AAV manufacturing project. The large number of design ideas was why Biogen chose to work with us. So again, this scale of data. Second, it's not just your data asset, right? So again, I will highlight the structure of the biotech industry today is that every company has their own pool of biotech IP, and they're not, even if it's, like for example, let's say you were a food protein company and you invented a better way to have high production of protein titers for your food protein. Well, some other cosmetics company that's also interested in protein production could use that IP. There's the friction to get that, get that data, those intellectual property from one company to the other today effectively means that that does not happen in the industry. And so one of the things we do at Ginkgo is we aggregate those learnings. We aggregate that data and IP, and we make it broadly available as a pool to anybody on the platform. And I am working my butt off to do more of this. We acquired Warp Drive Bio, Dutch DNA, Radiant Genomics, Circularis, Novogy, the Bayer Microbial Biologics Group up in West Sacramento, Lodo, Stride Bio. We're bringing in all these assets, not, again, I will highlight, to develop our own drugs or food products or anything else, but just so that you have access to them. All right, and you can look at the numbers on this, right? So if we do a like-for-like comparison of this is just one of our data assets, a microbial and metagenomic proprietary genome database. We have about 2 billion unique genes in there compared to more than 10-fold less in the public databases. That's, again, all available to you to search for novel proteins, natural products, industrial enzymes, things like that. And so, from Sumitomo, it was the great transparency and sophisticated data set that got them working with us. Okay. Next one, going quickly, all right? So really nice quote here from Nicholas Oler at Lygos, Chief Technology Officer there. The project team at Ginkgo is great, but the early results on one of our projects are stunning and supports Lygos' mission of accelerating the world's transition to high-performing, sustainable products. Okay, if you look at our press releases, this was a project that started like 6 to 9 months ago. So how do you get stunning results in biotechnology in 6 months? You don't start from scratch, right? So for the project we did from Lygos, we were able to leverage existing both biological assets and high throughput infrastructure that was already ready there on the shelf, ready to go, to drive that project more quickly, right? Again, being able to speed these things up is critical, right? Trent from Microba, SVP Therapeutics, Ginkgo's expertise and resources have moved our drug discovery project along at a pace that just would not be possible either using internal resources or via traditional CRO. Bob Ryder heads up R&D for crop sciences at Bayer, right? Open innovation will accelerate the product pipeline. We'll make sure that we bring high quality biological solutions to the market faster, right? So again, by outsourcing this technology at Bayer, it's a place that has a lot of resources, they thought they could move more quickly. All right. On that front, Jasmina, CEO at Archaea, spoke at, we have Ginkgo Ferment, our annual meeting. We did just about a month ago in Boston. And Jasmina got up on stage and she said, hey, I was up here a year and a half ago telling you about the launch of Archaea and that we had just did our fundraising round. And now I'm here up on stage a year and a half later and I'm telling you about our first product launch. Year and a half. Okay, that is very fast in biotechnology. It's a cosmetics company. I know it's not a drug, but still, it is fast, okay? And why is that? Well, normally when you raise, say, your $10 or $20 million kind of like A round or B round, what's the first thing you do? Oh, well, you call Alexandria Real Estate to get showed some overpriced lab space in Cambridge or South San Francisco. Then you call up your thermo rep to be shown some overpriced equipment that you're going to put into your lab and barely use. And then you start buying a bunch of consumables and hiring an R&D team. And $5 to $10 million later, you're ready to start doing work at scale. Huge amount of this precious cash that you're raising as a startup company is going into just creating the physical infrastructure to do the research work in the first place. Right? This has to stop. It has to stop. We should not be all building these low throughput labs independently. It is dramatically more efficient to centralize that work on automated infrastructure. We're trying to make that as easy as possible at Ginkgo. No pipettes. put down the pipettes. Okay. The other opportunity is around replacing fixed with variable costs. So this is this is sort of my chart of what an early stage pharma biotech looks like. So at the beginning, you would love to have as much R&D as you could possibly get your hands on. You're trying to develop a great drug candidate. Okay, so the more research you do, the better your candidate is. You want it to go fast because you're racing other people to get into clinical trials. So you want a lot of R&D. Okay, then animal trial starts, clinical trial start. Now you want like no R&D. You're trying to conserve your money, pay those clinical trials, get to the other side of some good results so that you can raise more money on good terms and then fund further development of your R&D pipeline. Well, what does your actual R&D spending look like? Oh, the dotted line in the middle. Because once again, you bought the lab, you got the equipment, you hired the R&D team, and they're just sitting there spending evenly across that period. So it undershoots at the beginning. When you wish you had more, it overshoots in the middle. Well, what happens in a tight capital market? I'll tell you right now, this is what's happening in Cambridge all over the place. In that little dip in the middle, the R&D team gets fired. Okay, not an efficient way to run our industry. All right, it would be more efficient if when you need a lot of R&D, you turn up the dial, you order more services. When you need less R&D, you turn it off. And again, centralized services, outsourced services allow you this type of control. I think it would drive more efficiency in general. So let's say you hear all that, okay, more R&D per dollar, you get to access other data sets, it's faster, lower CapEx, fixed with variable costs. Okay, I believe you, I want to do it, okay? How do you work with us? First off, there's a Ginkgo booth 10 feet out that door. Just please go talk to us, all right? We would love to talk to you about technical folks there. We can talk to you about your project. You can click this link at the top of our website, work with us, and you'll get follow-up from technical folks at Ginkgo again to hear your specific needs and talk about what our platform can do. But if you're like, that's too much work, I want to just know what you do, Ginkgo. Is there things you already do that are aligned with what I want? So towards that end, we started launching Ginkgo Services last year, our first service, Ginkgo Enzyme Services, just to get out there and show data about what we could do in the area of enzyme optimization and expression and so on. It was very well received. So again, at Ferment, about a month ago, we launched 4 new services: Ginkgo Microbe Services, Cell Therapy Services, AAV Services, RNA Therapeutic Services. And you can see some of the assets we've acquired that'll roll into that. Like, so for example, all those bare microbial assets in agriculture, that microbe collection, that all rolls into the microbe services. And I would encourage you to sign up. We have webinars. We'll show you all the details. We are an open book about the assets we have in these areas because we want you to make use of them. Okay. One other thing I want to tell you that I'm, again, trying to make this easier for you to use. So the number one request we've heard from customers talking to you all is to technically de-risk the work of cell engineering. In other words, this stuff just doesn't effing work sometimes, okay, right? And how do we make it so that it's more likely that a cell engineering project gets to success so that you can bring a product to market? And some people like already believe this, right? So, you know, Keith at Optimbia, like the likelihood of success for our project specifically was seen to be improved with the multifaceted technical capabilities at Ginkgo. Yeah, okay, great. You know, Nicholas at Lygos, as a CTO at a growing company, what could possibly do better than working with Ginkgo to de-risk my company's plans? Awesome, I love that, okay, but many other folks are like, BS, Ginkgo, right? Like nothing is predictable in biology. I don't care how many times you've done a project like this. My project is going to be different, and I don't trust that it's a high likelihood of success. Nothing you could say could make me believe that. I understand you, okay? So what I'm going to do is put our money where our mouth is, and we updated our pricing. So for 3 of our most mature services, specifically our enzyme discovery services, so you have a reaction, you want to find an enzyme to do it. Second, enzyme optimization, you want to make your enzyme better, so you know, higher specificity or productivity, and expression of proteins. Like you want to produce a protein at a higher titer. For those services, you tell us the technical specs. We'll tell you if we think it's nonsense. If we don't, it's payment on success. In other words, if we don't hit the spec, you don't pay. So you as a research head, you can either pay your internal R&D team, which if the project doesn't work, I assure you the money is still spent, or you could pay Ginkgo only in the event that we actually deliver what you want with that R&D project. Okay, so I'm trying to move from research to engineering via this success-based pricing. So expect us to be aggressive about this as more parts of our platform become predictable. Thank you, I appreciate the thumbs up. Okay, so what can Ginkgo do for you? It is on my shirt. So I just want to point out, we have a booth, it's 10 feet away. Please, if you heard all that and you're like, yeah, Jason, but if only you did this, then I would be interested in working with you. We want to know it. So we ended last quarter with $1.2 billion in the bank. I am investing that, again, not in therapeutics, not in food proteins, not in agricultural products. I do not have any products. I invest it exclusively in automation and IP, acquiring new data assets, whatever the hell you guys want. Okay, so tell us, it is your research budget, right? Like, what can we spend it on to make our services more useful to you? We are literally standing over there wearing shirts that are asking you to tell us. Okay, so please, please, please tell us. All right, I'm gonna end on this. Biology lives in the world of atoms, okay? I like Sam, I like OpenAI. You saw him testifying at the Senate last week, talking about the implications of artificial intelligence in the world of bits and what it's gonna mean for all of us. But ultimately, the world of atoms, like we're made of atoms, right? Food is made of atoms. Like synthetic biology is going to have a bigger impact and has bigger questions to answer on sort of ethics and impact than AI, and we need to start asking those questions now. And so I want to, again, give a round of thanks to SynBioBeta, who you'll see a number of sessions, yesterday, today, and tomorrow on these types of topics. I think it's really critical for all of us in this room to be asking these questions now, so when we're in front of the Senate 3 years from now, we have our shit together. Okay. All right, so towards that end, I'm going to end on this. I do like ChatGPT, so I asked it, ChatGPT, what's more important to society? Atoms, the world we all live in here in synthetic biology, or bits? It said, well, atoms and bits are both important to society, but they play different roles, have different implications. Atoms refer to the physical building blocks of the world. Bits refer to basic units of information. I was like, wow, this is really getting to be human-level intelligence. This is a complete dodge of my question, ChatGPT, right? So I said, okay, ChatGPT, but if you had to pick one, what is more important to society, atoms or bits? Well, if I had to pick one, I would say atoms are more important to society. It's because atoms are the building blocks of the physical world. They're essential for our survival and well-being. They make up the food we eat, the water we drink, the air we breathe. They make up the materials we build infrastructure for homes, roads, bridges. Without atoms, society would not be able to function in its current form. I could not have said it better myself, okay? Synthetic biology is the technology for programming in the world of atoms. We, I think, are really trying to build the platform that allow all of you to develop those applications that are going to make the world of atoms better. I cannot wait to see what you build with it, and please, please do go tell us what we can do to make that easier for you. Thank you so much for your time. Enjoy the rest of SynBioBeta.